Hello, dear students. In last lecture, we have also discussed about the IMVI classification, and by using that, we can also differentiate between the water contaminated with the fecal coliforms or not. So, in this regard, we have already discussed indole test and methyl red test. Now, the third test in row is nothing but the VP test, or we can say Voges Prosker test. And what is the principle behind this test is nothing but the this test is used to determine the capability of some organisms that to produce non-acidic or neutral end product like acetyl amyl carbinol from the organic acids which that result from the glucose metabolism. So it is nothing but the basic principle or mechanism behind this test. In last test, or we can say in MR test, we have already discussed. For the MR test, we can normally use MRUP broth, and for the VP test, we also use MRUP broth. And in this test, we have to determine the ability of that particular organism which is present in that particular water sample to produce non-acidic or neutral end product uh, like acetyl methyl carbinol after the incubation, so that some bacteria which can ferment carbohydrate into acids and 33% of carbohydrate into non-acidic end products for like 2,3-butanediol and acetyl methyl carbinol. So these are nothing but the non-acidic end product which can be synthesized by some of the organisms. As on the basis of acidic product and non-acidic product, we can determine whether the product which is synthesized by that organism we can uh, determined by addition of different indicators. For example, in indole test, we have added Covax reagent. For methyl test, methyl rate test, we have added methyl rate indicator. And again, also for VP test, we can also use another indicator, nothing but the Barrett's reagents, we can say. And if that non-acidic end product are synthesized by the organism in that particular media, we can determine by addition of that Barrett's region into that broth which is inoculated by the particular organisms, or we can say particular organism which we have inoculated in that particular media. This is an ordinary test to detect the presence of acetyl methyl carbinol. In short form, we can say it nothing but the AMC in the medium. The production of acetyl methyl carbinol can be detected whether it is produced by that organism is detected by adding zero point 0.5 ml of alpha naphthol and zero point 0.5 ml of Covax reagent, and that reagent are nothing but the we can other words we can say Barrett's reagent 1 and Barrett's reagent B. In case of Barrett's reagent A, it is nothing but the compose of zero point 0.5 ml, or that nothing but the compose of main constituent of that Barrett's reagent, nothing but the alpha naphthol, and another is Barrett's reagent B is nothing but the KOA solution, and we have to add zero point 0.5 ml of that particular Barrett's reagent into that broth after incubation. The acetyl methyl carbinol, if produced by the bacteria, oxidized by the KOH to diacetyl in presence of air, and that reaction can be uh, visualized by the formation of coffee red color. In that diagram, we can see it is nothing but the mechanism behind that test. Here we can say if that media contain glucose, after the metabolism of that particular organism, after incubation, we have to add uh, Barrett's reagent into that media. If there is a non-acidic end product or synthesis by that organism, which can be determined by the addition of the Barrett's reagent, and if the taste is positive given by that particular organism, that taste is visualized by like the formation of the coffee red color ring. And if the taste is negative, if they are not able to synthesize the non-acidic end product, the taste is remain as it is. So the taste is considered as a VP test negative, and another is VP test positive. In order to perform the VP test, an experimental organism which can be inoculated into MRP broth and incubated for 24 hours. Similar procedure for the methyl red test we can also use. After the incubation, a test reagent, that is Barrett's reagent A, added to the test tube, and then zero point 0.5 ml of Barrett's B reagent step we have to add to each tube, and we have to allow for 1 to 2 hour uh, in incubator, so that there is a formation reaction can be happen, and we can determine the coffee color formation in that particular taste if non-acidic end product are synthesized. So that taste is considered as a positive taste for the VP taste. So on that basis, if the formation of cherry colored ring in the case of indole taste, um, red color red color is formation in case of MR taste, and coffee color in case of VP taste, we can determine that taste are positive. In other words, if they are not found in that taste, we can see we can call it nothing but the taste are negative. So it is all about VP taste.